I'm Giulio DiDiodato. I'm one of the board members of Hospice Simcoe, and I was asked by Hospice Simcoe to provide an update on the COVID-19 vaccines, which is a very topical issue at this time. I'm also a critical care physician and the chief research scientist at the Royal Victoria Hospital. So I just wanted to put a disclaimer that whatever I'm going to talk about today is really they're my own views and opinions, and aren't necessarily the views and opinions of Hospice Simcoe or the Royal Victoria Regional Health Centre. Another disclaimer is that, there's been an extraordinary abundance of research papers that are either on their way to being published or have been published in the hundreds of thousands since COVID-19 has emerged. And so I just wanted to let you know that I'm not obviously a COVID-19 expert, and I find it hard to believe that anyone can actually be. So take it for whatever that's worth. As far as conflicts of interest, vaccines are a highly divisive issue sometimes. People have strong beliefs and opinions either for or against them. And I just want to let people know that regardless of what I say, I don't have any financial interests in or have ever been financially reimbursed by any of the vaccine makers of any of the approved vaccines. So for those of you who maybe don't know a lot about coronavirus, we'll give you a little bit of a brief introduction. So human coronaviruses are enveloped single-stranded RNA viruses. They're essentially 4 seasonal coronaviruses that circulate every winter. We call these endemic coronaviruses, and in the vast majority of people, they cause very mild upper respiratory tract infections. Some of you may remember back in 2002 and 2003, we had an epidemic coronavirus that emerged called SARS-CoV-1. It certainly sort of was much less significant in terms of its global impact than SARS-CoV-2, but nonetheless, it infected thousands of people, ended up killing about, I think, 770 people, and then just disappeared. As quickly as it came, it left. And we still don't understand why. There's another endemic virus called the Middle Eastern Respiratory Syndrome virus, another coronavirus that most commonly is found in the Middle East because one of its intermediary hosts are camels. It has a very high lethality rate, case fatality rates approaching 30%, but it's infected very few people, and it's mostly a sporadic illness. And then, of course, we have SARS-CoV-2, the pandemic virus that we're all living through now. And for those of you who want to get a sense of, what's the current status of SARS-CoV-2, you can see that there have been about over 76 million cases detected so far. There are obviously way more cases if you count those that haven't been detected or in those countries that are not reporting them all. And we've been able to ascribe to SARS-CoV-2 almost 2 million deaths, okay? And in Canada, there's been almost about 400 deaths per 1 million population. Now, do we really need a COVID-19 vaccine? Well, I guess the question is, what do we have available to us to try and curtail the impact that SARS-CoV-2 has had on our lives? Now, when we talk about viruses and their impact on populations, we frequently talk about a reproductive number. And the reproductive number is our way of measuring how infectious a virus is. Now, for SARS-CoV-2, we generally think that for every person that is infected, okay, they have the potential to infect on average, up to about 4 more people, who then, of course, can infect up to 4 more people, and they can infect up to 4 more people. So you can quickly see how this sort of process grows exponentially and can get out of control very quickly. Whenever we try to deal with this, with viruses and their infectious potential, we have a number of things that we can do to try and reduce this reproductive number to a very low number, at 1 or less, meaning we want to take the natural reproductive number and reduce it to 1 or less, because that essentially ends the chain of transmission for epidemics and pandemics. And so what do we have at our disposal? We have things like non-pharmaceutical interventions, things like wearing masks in public, social distancing, etc. We don't have a lot of drugs to prevent transmission with SARS-CoV-2. Obviously, vaccines play an important role in other viral illnesses, and seasonality obviously plays an important role in most viral illnesses. So if we look at the current state of affairs for Canada, right, we have about 525,000 infections, of which almost 15,000 have now died. So case fatality rate of around 2.7, 2.8%. And we're still seeing a lot of new cases every day, despite, essentially doing what everyone is aware of that we've been doing. And so we've put into place a number of non-pharmaceutical interventions in Ontario and in Canada that include limiting the size of social gatherings, mask wearing, closing some and now most businesses as we go into lockdown, obviously closing some schools and universities, and we haven't had a stay-at-home order. And when you look at the impact of non-pharmaceutical interventions, not only by themselves, but as a bundle, you can see that they take what is the reproductive number of SARS-CoV-2 at around 2.5 to 3, and can, in theory, bring the effective reproductive number down to about 1. So we should, with non-pharmaceutical interventions, if everybody was compliant, we should have had this sort of pandemic under control. But what do we see actually is the real case? Well, if we look at the estimation of the effective reproductive number over time for Canada, you can see when COVID entered our population and everyone was essentially immune naive and there were no non-pharmaceutical interventions, that is, no one was wearing masks and no one was social distancing, the reproductive number was around 3, which is what we know. But as we introduced non-pharmaceutical interventions, the reproductive number came down. Now, that should, in theory, then, we should see, of course, follow that lowering of the reproductive number, a decline in the number of cases. And initially, in the first phase of the illness, you could see that as the numbers initially rose and as the reproductive number was brought down by non-pharmaceutical interventions, that the cases also fell. Now, part of that also was due to seasonality. As you can see in the summer months, we had very low cases. Despite having reproductive numbers slightly over 1, the cases seem to be under control. But what you also see is, even though the effective reproductive number hasn't really changed very much, in fact, it's at its lowest, or at least we believe to be at its lowest now, you can see that the daily number of cases is increasing significantly and exponentially. And so there's a couple of things that are going wrong. One is we're either miscalculating the effective reproductive number, that is, our estimate is way too low, and in fact, maybe the effective reproductive number is much higher than we believe, which is almost certainly the case, right? Remember, the reproductive number is calculated by knowing how many secondary cases of COVID result from a primary case. Well, if we're in a situation where our public health agencies are completely overwhelmed and unable to do contact tracing, clearly we cannot account for all the secondary cases that arise from a primary case. And so we'll have a gross underestimate of the effective reproductive number, which is probably what's happening, because in theory, we should have this pandemic, at least in Canada, under control, given this effective reproductive number. And in fact, we don't here. So what else do we have? Well, if we're finding that our non-pharmaceutical interventions are not as effective as we would like for a variety of reasons, do we have any medications or other agents that we might use to try and reduce the transmissibility or the severity of disease? Here you see the entire list of all drugs and other interventions submitted to Health Canada for review and approval against COVID. So you have 1, 2, 3, 4, 5, 6, 7 agents that have been submitted to Health Canada for review and approval for COVID, 4 of which are vaccines, 1 of which is remdesivir, which is essentially a very expensive Tylenol tablet. We have 1 drug here, a monoclonal antibody that's supposed to be used in outpatients to try and prevent the progression of disease to severe disease. And then you have 2 vaccines, the Pfizer vaccine, the Moderna vaccine, both of which have been authorized, essentially under emergency authorization. You have 2 more vaccines, AstraZeneca and the Janssen vaccine, which are both under review, have not been authorized for approval. So you can see that the armamentarium that we have in terms of combating the current pandemic is very, very limited. Our non-pharmaceutical interventions are not doing what we want them to do or expect them to do. And then the drugs and other interventions we have are not very abundant. And so we've got a problem. And so what do? Well, we turn to vaccines. So let's just quickly review what happens and how coronavirus causes disease. So coronavirus is as I said, an RNA virus that has on its surface these proteins called spike proteins. And these spike proteins have a receptor binding domain, meaning a part of them makes a little connection with other receptors on human cells in our nose, in the back of our throat, and in the lining of our lungs with a specific receptor. And this receptor is fooled by the spike protein on the coronavirus, and it causes an internalization of the virus into that cell. Now, viruses can't replicate without hijacking the machinery of cells to replicate for them. And that's exactly what the coronavirus does. It tricks our cells into taking over the job of replicating all of the proteins and components that it needs to make other little baby coronaviruses. And those baby coronaviruses then cause lots of disruption of cells. They can escape and invade more cells. Now, we've been blessed with an immune system that helps combat pathogens such as coronavirus. So these coronaviruses are recognized by our immune system. And after they're recognized, essentially 2 things happen. We develop antibodies to specific parts of the coronavirus, and we develop cells, immune cells, that also have the ability to directly attack infected cells or the virus itself to combat the infection. Now, vaccines use our immune system, or at least prime our immune system, to fight infection before a native infection actually occurs. And so what we find is that there are many types of vaccines that are currently being tested for their effectiveness against coronavirus. There are virus vaccines. These are typically the types of vaccines that we get, for example, for influenza, an inactivated virus that we get an injection of, or the nasal spray vaccine, a live attenuated virus that does replicate within the body. The vaccines that we're going to talk a little bit about today are of the viral vector vaccine type. So they're non-replicating viral vectors. And this is the Oxford, AstraZeneca, and the Johnson & Johnson vaccine are of this type, where the coronavirus itself has been inactivated, meaning that the genes it needs to help replicate itself have been inactivated, but the spike protein gene is kept intact. And so when it enters the cell, it primes the immune system to create antibodies and immune cells directly against the spike protein in hopes that it can prevent the initial infectious phase. The other types of vaccines we're going to talk about today are nucleic acid vaccines, and specifically RNA vaccines. These are the Pfizer and Moderna vaccines. Now, Up to this point in time, there's actually been no previous licensed nucleic acid vaccines. However, they appear to be safe as far as we can tell. The RNA of the vaccines, the genetic material that these vaccines bring into the body, is not integrated into the human genome. The expression, that is the making of viral proteins from this piece of RNA, is very transient, meaning it stops after a certain period of time. And the mRNA that's actually injected is metabolized and eliminated by natural cellular processes. And the real advantage of these vaccines is they're very flexible, because you only need enough genetic material to code for the part of the protein that you want expressed. And if mutations arise, and there's lots of talk about mutant coronavirus species, you can always modify the piece of RNA that's used in the vaccine to create a protein that more closely mimics the mutant version of the protein in these new coronavirus species. So just a quick overview, give you a sense of how active the vaccine community has been in trying to develop coronavirus vaccines. As of today, there are 61 vaccines in clinical development, meaning they're being tested in human beings, and 172 vaccines in preclinical development. Okay, so a lot of work is being done, even though we already have several vaccines that have been approved and several others that are imminently going to be approved. For those of you who want a brief history of the benefits of vaccines, I don't think that anyone who's rational can ever deny that the human health impact of vaccines can't be overstated. It's estimated that they save about 6 million lives per year. That equates to 360 million life years saved. They've been responsible for the eradication of smallpox and rinderpest. Don't forget that smallpox sort of throughout human history has killed about 60 million people. And polio is on the verge of also being completely eradicated, remaining in only 2 countries, Pakistan and Afghanistan. Certainly they have indirect benefits, meaning that by my immunizing myself, I potentially can provide protection to other people who may not be able to be recipients of vaccines. So it might help provide protections for immunocompromised, elderly, marginalized populations, pregnant women, neonates, and young children, again, who may not be eligible for vaccination. Certainly, we are starting to see their emerging role in cancer prevention, with prevention of liver cancer through hepatitis B vaccines, and a prevention of cervical cancers through human papillomavirus vaccines. And they're extraordinarily cost effective, estimated to cost about $50 for every life saved. Now, ideally, it would be great if within one vaccine, we could have it have an impact on all aspects of the coronavirus infectious phases. In other words, it would be fantastic if one vaccine could be shown to prevent infection, either the initial infection in an individual or reducing the probability of transmitting the disease to others. It would also be fantastic if you did develop disease, if the disease that you developed would be super mild, so it didn't progress to needing hospitalization or death. Today, we're not sure that any of the vaccines that we have do all of these things. And the part of the problem is that when these vaccine trials, these human trials were set up, if you look at what were they set up to be able to detect, or what were the endpoints that we were going to evaluate to assess whether the vaccines were effective or not, that really the only endpoint, at least in the initial phases of testing, were to see if it could prevent symptomatic disease in the recipient. They were not set up initially to see if they could reduce severe disease, either hospitalization or death, or if they could be preventative, that is, interrupt either the initial infection or the transmission of infection to other people. So as of today, we don't believe that no vaccine's been shown to demonstrate sterilizing immunity of the upper airway. In other words, there's no vaccine that we know that's caused our immune system to develop resistance to initial infection in our nose and throat. And we don't know what the immunological correlates of protective immunity are. In other words, we haven't really done blood tests on our, for example, antibody levels or our immune cell levels to know, well, if we develop this amount of antibody or if we see these types of immune cells, then we can say by definition that we are immune because we have the clinical correlate to compare it against. Now, the WHO set some target profiles for the development of COVID-19 vaccines, and these were the minimally acceptable criteria that they believed any vaccine should have for it to be licensed and to be effective against SARS-CoV-2. So they certainly believe that every vaccine would have to be used in conjunction with other non-pharmacological interventions. And we'll talk about why that's the case in terms of vaccines effectiveness. They should be effective in all adults, including the elderly, but there was some acceptance that there might be populations for which these vaccines might not work at it, including the immunocompromised. While there was an acknowledgement that almost certainly we would have reactions to them, it was felt that none of them should be severe. And the minimum amount of efficacy, that is, how much should they reduce disease by, was to be believed to be at least 50%. And the reason for that is if we wanted to have any chance of developing herd immunity, then we needed to have vaccine efficacy of at least 50% that lasted for at least 6 months after the final booster dose of vaccination. And that the endpoints that we were talking about in terms of important endpoints was that the vaccine should reduce symptomatic disease, they should reduce severe disease, and they should also, if possible, prevent disease. Ideally, there would only need to be 1 dose, but 2 doses was felt to be acceptable. However, they felt that if you needed to have repeated dosing, that it should not be needed for less than a 1-year interval. So if you needed to immunize people like every 6 months, they felt that that would not be not only a good vaccine, but probably not an acceptable vaccine to the population. So when we talk about vaccine efficacy, what do we mean? That means it's the proportional reduction in endpoint between vaccinated people and people who don't receive the vaccine or are in a placebo group. And generally what we do is a randomized controlled trial. So people volunteer, they get allocated in a random way to either receiving the vaccine or some control. And this type of effect is really the best case scenario for the direct benefit of the vaccine in the individual on the endpoints that we talked about. But in reality, when we go then and administer the vaccine across the population, we measure the vaccine effectiveness. And the vaccine effectiveness really is what is the protection attributable to the vaccine against SARS-CoV-2 when it's administered in the real world. And it includes both the direct and indirect effects on the population, or what we call herd immunity. But almost always, effectiveness is less than efficacy. So efficacy generally gives us the best case scenario. Now, there's lots of challenges you might imagine when you're trying to do a vaccine trial in the midst of a pandemic. So obviously, there are differences in the natural exposure of people in the study to COVID-19 in terms of, for example, if you're a high-risk person as a healthcare worker or maybe living in a shared environment. And then also what is the variable compliance with non-pharmaceutical interventions, because as we'll see, the effectiveness of the vaccine is dependent on the use of non-pharmaceutical interventions. How many people do you enroll in the trial? It's always hard to know how many events you'll need to determine a difference between a vaccine and a placebo group. What are the patient characteristics? We know that there are certain races and cultural marginalized populations, those with comorbidities and immunocompromised, those who are socioeconomically disadvantaged, who seem to have much worse outcomes with SARS-CoV-2 infection than other in the population. And so you want to make sure that your enrolled population is representative of these groups. We also know that healthcare systems around the world vary in the quality of care that they bring. And so the outcomes may be affected not by the vaccine efficacy, but the quality of care that people receive. And then there's the duration of observation after vaccination. What you'll see today is that the initial results and the initial approval of both the Pfizer, of Moderna, of the Oxford AstraZeneca vaccine are literally based on 2 months of observation after the final dose, right? So we're talking about very short-term observation, and we're looking really for safety signals. So immediate adverse effects and reactogenicity. And then we're also looking for vaccine efficacy against symptomatic disease only. And if we're lucky, maybe we might see a signal that it might reduce severe disease, but highly unlikely. Longer term, because these studies are meant to last for a year to 2 years, we're really looking at overall vaccine safety, looking for very rare events that we would only capture when millions of people are vaccinated. How long does the actual protective immunity last? So right now we have perhaps 2 months of protective immunity under observation, but we really want to know, is it 6 months, 12 months, 2 years? And then we want to be able to detect, if we can, is it efficacious against not just disease modifying endpoints, but against endpoints for prevention, meaning reducing infection and reducing transmission. So here are the results for 3 of the vaccines, and I only did these 3 because none of the results for the Janssen, Johnson & Johnson are publicly available, and even the Oxford AstraZeneca are only available through a paper. But the Pfizer and the Moderna vaccines, through their FDA submission, had a very robust data that was available to the public and is available for you to review if you want to review it yourself. So let's look at the type of vaccine. As we said, that the Pfizer and the Moderna are the mRNA or nucleic acid vaccines. The Oxford AstraZeneca is an inactivated virus vector that contains the gene for S-glycoprotein. That's the target, the antigen target. That is the target for our immune system, is the spike glycoprotein, okay? All 3 of these vaccines require 2 doses. There's 30 micrograms of material in the Pfizer, 100 micrograms of material in the Moderna, and there's this many virus particles in the Oxford AstraZeneca, although they gave 2 doses in their study. All of the modes of delivery are intramuscular. The Pfizer second dose comes 21 days after the first, whereas for both Moderna and Oxford, 28 days is the time period between the first and second dose. Obviously, these are large studies involving tens of thousands of people. The Pfizer was the least restrictive of the studies, including even children who were 12 years and older. They had no restrictions in terms of your previous COVID-19 status. They didn't care whether you were previously infected or not. And the rationale for that was obvious, is that they presume that in the real world, we're not going to test people to see if they've been previously infected with COVID before we give them the vaccine. In fact, we're probably not even going to ask. We're just going to vaccinate everybody. Whereas Moderna and Oxford, in fact, excluded people who had had previous infection with COVID vaccine. And their rationale was, well, they wanted to see the true effect of the vaccine in COVID naive patients. The Pfizer vaccine was done in many countries, which is important because you want to be able to see the effect of the vaccine in different contexts. The Moderna vaccine the study was done entirely within the US, and the Oxford AstraZeneca was done in 3 countries. However, the results that they reported initially are only from the UK and Brazil. The sort of breakdown of the types of gender, race, and ethnicity that was included, in general, there was a good split between males and females. The vast majority of enrolled patients were white in all studies. And when you see the Black sort of enrollment of non-Caucasians, 9.8% of the Black population in Moderna and Pfizer, and even less in Oxford AstraZeneca. The representation of an older population, which is obviously very important, given the exponential increase in mortality that's associated with age. So 21% of people were over the age of 65, and about 25% in the Pfizer and Moderna studies, respectively. The Oxford AstraZeneca was predominantly done in healthcare workers, so a much younger population. And then you see people with comorbidities, which again, we know increases the rate of death in people infected with COVID. When you look at the endpoints, the reason that they chose to look at the rate of infection after the second dose is from their phase 1 studies. And phase 1 meaning they took a bunch of healthy volunteers and gave them different doses of the vaccine. So this is the phase 1 study for the Pfizer vaccine. So for example, in this group, they got 10 micrograms of the vaccine. In the second group, they got 30 micrograms, which is ultimately the dose they used in the study. And then another group, they got 100 micrograms. And they compared what they called neutralizing activity against the serum from people who had were proven to have been infected with COVID-19 previously. And this really means that they allow, in a cell culture, they have human cells, they put virus in that human culture, and they see how much of the human cells get destroyed. And they call that this 50% neutralizing titer. So the fewer human cells that get destroyed, the more, quote, neutralizing activity. The presumption is that there's something preventing cells from virus from entering those cells and killing and destroying those cells. And so you can see that these are the days where the viruses were administered. So the first dose administered on day 1, the second dose administered on day 21. And you can see that after the first dose, there's not a lot of neutralizing activity, regardless of the dose that you use. It's only when you get to day 28 after the first dose that you really start to see a lot of neutralizing activity that almost exceeds the neutralizing antibody activity seen in people's, the serum of people who have been previously infected. So that's why when they looked at endpoints, they picked these endpoints. They started counting, if you will, the number of symptomatic people with COVID 7 days or greater after the second dose. It was 14 days or greater after the second dose in the Moderna, and same with the Oxford AstraZeneca. The full duration of the trials are supposed to be about 2 years for both Pfizer and Moderna, and a year for Oxford AstraZeneca. So obviously, the reports that you've seen either publicly or in journal articles really represent only the first 2 months of observation after the second dose has been administered. So what did they find? Well, in this 2-month observation period, you can see essentially the difference between the vaccine and control groups in terms of the number of symptomatic COVID illnesses. So for example, in the Pfizer, they detected 9 cases of COVID, symptomatic COVID, amongst 21,669 participants versus 172 symptomatic COVID cases in the control group. So that translated into a vaccine efficacy of 94.8%, meaning the proportional reduction of disease in people who are vaccinated compared to the controls. So a very efficacious vaccine. When they looked at the proportional reduction in symptomatic COVID in those over 65, what they found again was that the vaccine efficacy was about the same as in the entire group, which was really positive because, of course, there was this concern that the vaccine would not be as effective in an elderly group compared to a younger group. But that didn't appear to be the case in either the Pfizer or the Moderna. For Oxford AstraZeneca, this data was not reported. It was actually not done. For severe COVID, now remember, in the first 2 months, it'd be highly unlikely for us to be able to detect a signal in death or reduction in death or hospitalization. You can see how few cases they actually saw. And so, you really can't say very much about whether these vaccines are going to be effective against reducing severe COVID disease. as far as the safety data go, I think it already strikes your eye when you look at injection site reactions, especially for both the Moderna and Pfizer. The majority of people had reactions. Most of them had pain. Almost all of them had swelling and redness. Okay, but almost none of them were really severe. Same with systemic reactions. The vast majority of people had fever and fatigue. Okay, but again, when you look at the serious adverse events, which is what we're really concerned about, there was really no difference between the two groups, vaccine and control, across all three studies. And if you looked at mortality, that is, were there more deaths in the Pfizer, in the vaccine versus control, again, there was not a significant difference in the number of deaths. Just some vaccine characteristics. You all heard about the issues associated with the Pfizer vaccine in terms of its cold storage requirements. It has to be stored at -60 to -80 degrees Celsius, which makes its distribution obviously very logistically challenging. But once you thaw it, you have about 6 hours to administer it. Moderna, while it has some cold storage requirements, it can be stored at more common refrigerator temperatures for up to 30 days. So logistically, its distribution is much easier. Same is true for the Oxford AstraZeneca vaccine. So then, what does this all mean? Well, we have to really ask ourselves, well, how many people and how often would they need to be vaccinated to both end and prevent the pandemic? Well, this is really complicated, but it does depend on a variety of things. And in this particular model, we looked at that it depends both on the vaccine efficacy and the duration of protection, protective immunity. So for example, we know in all circumstances that more people would need to be vaccinated in year one compared to subsequent years. So if you had a vaccine efficacy of 90%, as Moderna and Pfizer claim to have, and we find out 10 months from now that the duration of their protective immunity is up to a year, we'd still need to immunize about 90% of the population to essentially end the pandemic, okay? Year two would only require about 50%. So it's not an insignificant effort required not only on those who are responsible for the manufacture and logistical distribution of the vaccine, but also trying to convince 90% of the population to actually be vaccinated. Now in Canada, if you look at the target populations for COVID-19 vaccination, for example, if we wanted to vaccinate enough people to maintain our essential core services, here's the numbers that we would have to immunize in thousands. So for example, healthcare workers, physicians, nurses, and midwives, and this is not counting all of the other healthcare workers who may not necessarily be patient-facing or may find themselves in alternative healthcare facilities, there's at least 463,000 of us, right? So if you're using a Pfizer vaccine, you're going to need close to over 900,000 doses. For police and military, there's 141,000 you'd have to immunize. And then for those people working in what we call essential services, there's over 2 million people, okay? If you're looking at the group of people who are in the tier 1 group in Canada, for example, those aged over 80, there's 310,000 of those people that we would have to immunize, okay? If you look at anyone over the age of 60 with at least one underlying condition, over 6 million people. And here we'd be immunizing these groups to try and prevent severe COVID disease. If we wanted to try and prevent symptomatic infections and reduce transmission, you're looking at a healthy population, okay? But look at the millions of people that we would have to immunize to be able to achieve that goal. Now in Canada, as you all know, the priority 1 group includes the residents and staff of senior living settings, those over the age of 70, healthcare workers with direct patient contact, and adults in Indigenous communities. Those are the tier 1, priority 1 groups in Canada. The second priority groups are the remaining healthcare workers not covered in the first wave, residents and staff of all other shared settings like prisons, homeless shelters, boarding houses, migrant workers, and then all other essential workers. Now, what is the status of our global vaccine production? I mean, we talk about immunizing these people and the effects we would expect from immunization, but logistically, I mean, is it even possible? So this is sort of the global vaccine production that's estimated to be available by the end of 2021. And these are the millions of doses that may be available. So for example, AstraZeneca is claiming that they may be able to manufacture 2.5 billion doses by the end of 2021. The only vaccine manufacturing capacity we have in Canada is with Medicago, and they're presuming to produce about 100 million doses of vaccine. And then you kind of see for the vaccines that are approved or close to approval in Canada, where the production is occurring. Oxford AstraZeneca has hedged its bets by producing that vaccine in 7 countries, but places like Pfizer and Moderna are only producing vaccines in 2 countries. So if something happens to either the supply chain or the manufacturing capacity in one country, they may be in real trouble in terms of being able to manufacture what they're predicting to manufacture in terms of volume. As far as countries pre-ordering vaccines, and this was going on ahead of time, Canada by far and away is the greediest country of any country in the world. It has pre-ordered about 9 doses per capita of vaccine, okay? And it also has the option of pre-ordering 2 more additional doses. So Canada's received a lot of criticism for doing this in the sense of hoarding vaccines from other less wealthy countries. Of the vaccines that Canada has pre-ordered, they've pre-ordered about 0.53 doses of the Oxford AstraZeneca vaccine, about 2 doses of the Pfizer BioNTech vaccine, 1.5 doses of the Moderna, 1 dose of the Johnson & Johnson, about 2 doses per capita of the Novavax, which is in a phase 3 trial right now, almost 2 doses of the Sanofi GlaxoSmithKline, which is in phase 1/2 trial, so a long way to go, and then options for ordering more from these other companies. Now, given how much pre-orders they've had and the assumed production capacity, supply chain security, and logistical distribution capacity, there was a suggestion that healthcare workers and high-risk population in Canada, in theory, should be fully immunized, enough to achieve herd immunity, maybe 60 to 70%, by April of 2021. And they're predicting that maybe by July, that they may see enough of the population immunized to achieve herd immunity. But we're going to talk about whether this kind of forecast is realistic. If any of you are interested in the cost, or if you've bought stock in any of these companies, certainly the most expensive vaccine is Moderna. It comes at $37 US a dose, and you need 2 doses, so over about $75 a dose. The Oxford AstraZeneca is certainly by far and away the cheapest vaccine, and will probably be the vaccine that's most commonly used globally. What about other vaccine-related issues? Well, how long will immunity last? What about post-marketing surveillance for safety? We've heard about these new SARS-CoV genetic variants. Well, will they have an impact on vaccine effectiveness? There's a phenomenon known as antibody-dependent enhancement of COVID-19 disease. Will this come into play in terms of vaccinated people? We don't know. And vaccine effectiveness during a pandemic. So let's talk about reinfection, and which relates to obviously the duration of immunity. Now, the way we've defined reinfection in the literature, and there are many definitions, but basically, no matter what definition you use, there have only been about up to 100 case reports worldwide. And almost all of the infections occurred within 150 days from the first infections. And almost in every case, every secondary infection was less severe than the primary case. So reinfections occur, and it brings into doubt in some people that they have developed enough protective immunity to protect them from secondary infections. Almost certainly reinfections are underreported. So there are probably way more reinfections that are reported in the literature. And what this means for us is that maybe social distancing, mask precautions, and other non-pharmaceutical interventions may still be needed even after a primary infection. And if reinfections are common, that is, protective immunity doesn't last a long time, herd immunity may not be achievable, okay? And also suggests that even previously infected people should, in theory, then be vaccinated. For how do we develop an immune response to SARS-CoV? Well, viruses that infect our mucosal surfaces, which is what SARS-CoV-2 does, and don't enter the bloodstream, generally result in antibody responses that are present for months to years. We develop neutralizing antibodies to SARS-CoV-2, that is, antibodies that prevent the initial infection. We develop more antibodies the more severe an infection is. So for those people who develop asymptomatic infections, they simply may not have protective antibody levels. But these may not correlate with the amount of antibody present at the site of infection, okay? So the blood levels of antibodies may not represent how protected we are from being infected initially in our nose and throats. The blood levels of antibodies appear to be present for up to 6 months, but we can certainly see they start to decline 1 month post-infection. However, we do develop these memory B cells, which are the producers of our antibodies, and they certainly are present for longer than 6 months. And they may have protection against symptomatic disease and severe disease. We also develop cell-mediated immunity, and this certainly is present for longer than 6 months. And this immunity may be especially important to prevent the initial infection at mucosal surfaces. As far as post-marketing surveillance, we have a pretty robust passive and active mechanism for reporting and monitoring severe adverse effects after vaccination. I'm not going to go through it, but basically every country has a system for this kind of monitoring. In Canada, the responsibility is shared between Health Canada and the Public Health Agency of Canada. Reporting of adverse effects following immunization is mandatory to Health Canada by healthcare providers or immunization providers, but these has to be reported by the patient to these agents. And Canada is also collaborating with global partners, such as the World Health Organization, in case there are other adverse events occurring in other places that may not be occurring in Canada, but we need to be aware of them. Now let's talk a little bit about this genetic diversity of SARS-CoV-2. Now there's a concern that vaccines that we currently have in development may not be as effective if we see SARS-CoV-2 variants arise that are genetically dissimilar from the virus that's used to develop the vaccines and our immune response. The one advantage that coronaviruses have, unlike other RNA viruses, which you see here, so here are the family of human coronaviruses. These are other RNA viruses such as mumps, hepatitis B, measles, and then things like HIV, influenza, and HCV. And think of the size of these bubbles as representing the amount of genetic diversity within each of these viruses. Coronaviruses are very unique because they have very large genomes. Their genomes encode for a lot of proteins. And one of the most important proteins they code for is one that corrects all kinds of replication errors that happen while the virus RNA is being reproduced. So the mutation rate in coronaviruses is about 10-fold lower than would be expected amongst these many other types of RNA viruses. And also, there hasn't really been a lot of time for coronaviruses to develop and accumulate a lot of mutations, especially with the spike glycoprotein. And so you see, when you look at the genetic diversity of coronavirus, it's tiny compared to all of these viruses, these other viruses, okay? And up to this point in time, there's no evidence that any of the reinfections that happen in human beings are either due to these mutant coronaviruses evading our immune system, okay? So what you've been hearing about in the news lately in terms of the spread of this one particular coronavirus mutant is this D614G spike glycoprotein variant that everyone keeps talking about, originated in the UK. It didn't originate in the UK. It's been around since the beginning of the detection of coronavirus in humans. And it's believed that more likely what's happened is that this particular clone is just spread globally because it seems to be a bit more infectious than other mutants for coronavirus. Just be aware that there are about 4,000 documented mutations already in the genomic libraries for coronavirus. So this is just one mutation. As I said, it appears to increase the infectivity, but not the severity of disease. And so it's unlikely that this is going to impact vaccine efficacy. First of all, this mutation is not located in the receptor binding portion of the spike glycoprotein, which is where we believe our immune system attacks. And we've also demonstrated in vitro neutralization assays, demonstrated that if you take the serum of someone who was infected with the native coronavirus, that serum protects against infection with the mutant coronavirus. So it appears to still be recognizable by the neutralizing antibodies of our immune system. Now, there's this weird phenomenon called vaccine-related antibody-dependent enhancement, and it was initially recognized in the 1970s with dengue illness. But basically what this is if a person has either low amounts of neutralizing antibodies or neutralizing antibodies that don't strongly adhere to the spike glycoprotein, then what happens is these antibodies actually promote the uptake of virus into the human cells via a different receptor and can actually result in much more severe disease. We are developing vaccines that have the parts of the spike protein that should result in the formation or the promotion of the best neutralizing antibodies. So we don't think that this will be an issue in those who are vaccinated. And we certainly haven't seen a signal of antibody-dependent enhancement in the people who are currently enrolled in these vaccine studies. So it may happen as we have more and more people vaccinated, but we haven't actually seen a signal yet. Now, vaccine effectiveness during a pandemic. What are the determinants that affect vaccine effectiveness? Well, vaccine efficacy may have an impact, as we discussed, on disease prevention, disease modification, but ideally we would love to have a vaccine that affected both of these, okay? What also determines how effective a vaccine is in the real world is our manufacturing capacity and distribution. And that's defined as how much of our population can be vaccinated in a day. Then there's also the issue of coverage, which is what is the uptake of vaccine by the population? We define this as the cumulative proportion of the population that's ultimately vaccinated. And then the phase of a pandemic we're in has a profound impact on how effective a vaccine is. So better adherence to non-pharmacological interventions will actually improve the effectiveness of a vaccine. Well, if you look at the most recent poll amongst Canadians of how willing are they to be vaccinated, you can see about half of them say, yeah, absolutely, give me the vaccine right now and I'll get vaccinated. Another 30% say, well, I'd like to wait a little bit, but yes, I'm very open to it. So about 70% of the population appear to be willing to get vaccinated. The most common reason, of course, for those who don't want to be vaccinated is they're worried about the long-term side effects that we don't simply know about yet, and that's fair. But here's, I think, maybe the most important slide that I have in the talk, and this looks at how is the effectiveness of the vaccine impacted depending on what's happening during a pandemic. So if you look at down in this area here we have a scenario where the effective reproductive number is 2.1, which is probably a more realistic reproductive number that's going on in Canada right now than the effective reproductive number of 1 that I showed you in the initial slides. And then you can see like whether you have a vaccine that's effective against, that prevents infection or transmission, that modifies disease, or is a composite of both. And then you look at the scenarios. And here you have 0.5% pace, meaning 0.5% of the population is vaccinated every day. That means 185,000 Canadians would have to be vaccinated every day. And we assume that 50% of Canadians will actually ultimately be vaccinated. And then you look at the vaccine efficacy. So 25% is dark blue and up to 90% is light blue. And you can see that in the midst of a pandemic with an effective reproductive number of 2.1, even if you could vaccinate 185,000 Canadians a day and 50% of them with a 90% effective vaccine, you'll only be able to reduce infections by about 40%, right? So the important thing is, if you can somehow reduce that effective reproductive number from 2.1 to 1.5 through things like ongoing non-pharmaceutical intervention compliance, you get much, much higher rates of reduction in infections with exactly the same vaccine, okay? Exactly the same pace of vaccination and exactly the same coverage. So it just reinforces the importance that for those of you who believe that as long as we get vaccinated, we'll be able to sort of completely forget about being compliant with non-pharmaceutical interventions, that's not the case. The vaccine will only be as effective as we are compliant with non-pharmaceutical interventions. The two are completely tied together, okay? So if you, as I said, if you look at that, a 0.5% pace is 185,000 Canadians a day. So if we wanted to achieve 70% coverage, so if those 70% of Canadians actually were willing to get vaccinated with a vaccine of 90% efficacy, and we know that it takes 35 days to develop protective immunity after immunization, the days to achieve this goal is 175 days or 6 months. So, you know, we're looking well into late spring, early summer, even to achieve this goal. Now, if you want to see, well, what are our vaccine, like how are we doing so far with, of course, our limited supply, okay? You can see here, here's the daily number of Canadians who've been vaccinated so far. So for example, yesterday, we were able to vaccinate 5,356 patients, the most people. The most we've vaccinated so far is 6,375, right? So remember what I told you, a 5% pace, a 0.5% pace is 185,000 Canadians a day. We have a long, long way to go before we vaccinate people at a rapid enough pace and at a significant enough coverage before we ever see a period of time where we can start to relax the non-pharmaceutical interventions. So what do you want from a COVID-19 vaccine? Well, I think everybody would say we want to return to normal, right? But built into that, we need a safe vaccine. We want to minimize the number of injections, both in terms of how many you get up front and how often you get them. We want the efficacy to be very high, and we want to minimize sort of social disruption by ensuring that we're compliant with non-pharmaceutical interventions in the short term to try and get the pandemic under control and maximize the effectiveness of the vaccine, with the view in the long run that the vaccine will help us ultimately minimize how much non-pharmaceutical interventions we need to continue to use on a long-term basis. So thank you very much. And I don't know if there's time for questions, but I'm happy to entertain any questions that people have.
Hey, Giulio, it's Chris Martin. Can you hear me okay?
Yep.
That was amazing talk. I learned a ton. Thank you. What's the normal period of observing effects from a vaccine before it's approved? This one was 2 months.
Usually a vaccine would go right to the very full end of the study. So generally speaking, you saw the duration of the planned duration of those studies were 24, 26, and 12 months amongst Moderna, Pfizer, and Oxford, respectively. It would come at the end of that period.
Thanks.
So Giulio, I'm sorry, with the math there, if you're for the amount of doses that we need to do, how long are you thinking then? That are we, like, it's not possible, I don't think, to do this by the spring. I've heard the government is saying approximately, you know, by September, the majority of the population will be immunized. Those who want it will be immunized by September. Do you think that that is going to be an effective amount of people?
Well, it depends how many of those. Remember that there are a lot of things that have to go right? That the supply chain can't be interrupted, the manufacturing process can't be contaminated, that logistically we can distribute these vaccines to those who want them. that enough Canadians are willing to be vaccinated, that no SARS-CoV-2 virus mutant emerges that can actually evade the protective immunity conferred by vaccine, but not against itself. We don't know what the duration of immunization is. For example, are the people who are immunized yesterday going to need another shot in the summer because we find out that protective immunity only lasts 6 months? So there's so many ifs to the question that you're asking. I don't think anybody can realistically answer it.
Okay. Thank you. Are there any other questions? I think you just have to unmute yourself. Okay, well, it's 1:03. So, oh, Lori's coming to the, do you have a question, Lori? Oh, she does. Okay.
I'm sorry, I have a question as well.
Okay.
I just wanted to say a big thank you from us here in the room. That was excellent.
You're welcome. Thank you.
Hi there, my name is Josie. I have just a quick question and I'm not really sure if you're able to answer it fully, but I know there's a lot of question in terms of the vaccine for those who are considering getting pregnant or wanting children in the future. Is there any information that you have in terms of how that would affect somebody who is wanting children, or even if they get pregnant after they get the first dose and then possibly can't get a second?
Yeah, so that happened. I mean, as you might imagine, that happened in the vaccine trials that were published. So all three, the Pfizer, the Moderna, and the Oxford AstraZeneca, excluded pregnant women, and they demanded that they be on some contraception. But you know, no contraception is perfect, and if you're going to have 40,000 people in a study, guess what? You're going to have a bunch of pregnant women who received the vaccine. So, but it's a handful, right? So there are women who subsequently became pregnant after receiving the vaccine dose, the first vaccine dose. They clearly weren't eligible to receive the second vaccine dose. As far as we know, for the 2-month observation period, you know, nothing has gone wrong, but who's to say that something won't emerge later on? You know, it frankly depends on the technology too, right? I mean, we don't have any previous nucleic acid vaccines, so it's hard to comment even historically on other vaccines that might resemble the SARS-CoV-2 vaccine to say, well, this should be safe because we use this virus technology, right? So I think more important is not so much that it's a coronavirus vaccine, is what type of vaccine is it? If it resembles, for example, inactivated virus vaccines like the influenza vaccine, there's really no reason to believe that that type of coronavirus vaccine would somehow impose some different risk to pregnant women and should be safe, right? Because we know that that inactivated virus influenza vaccine is safe in pregnant women. So more important is not it's a coronavirus vaccine, it's what type of vaccine technology was used to develop that particular vaccine.
Thank you so much.
Okay. Well, there's lots of little things being said in the chat. Thank you, very interesting, much appreciated. Asking people, you know, they want to share this video with their secondary school kids and sharing it with friends and relatives. So Hospice has plans on posting this to our YouTube channel. So it should be up there in the next couple of days, but
Can I ask a question?
sure.
Okay, sorry.
Yeah.
So it's Carolyn, and I'm just wondering, so if we don't get to the point of this vaccine being effective, are you laughing at me? Anyway, sorry.
Oh, I'm coughing. I'm sorry. I'm coughing.
Okay. Oh, sorry. So what does the future hold then for the population? Like if this vaccine doesn't work, if people aren't willing to have the vaccine, what's going to happen? Or do you have an opinion of what's going to happen?
It's a
Is
great
it gonna,
question.
is
Yeah.
it gonna peter out? Is it gonna be a seasonal thing like influenza or?
I mean, I think it's hard to believe that. I mean, it clearly has behaved differently than SARS-CoV-1, right? SARS-CoV-1 just
Right.
disappeared. It hasn't done that. It lacked, I mean, while it had a seasonal pattern, it still was very active even in the summer. So unlike the human seasonal coronaviruses, the endemic ones, which essentially disappeared during the summer, it didn't do that either. It hasn't done what MERS-CoV does in the Middle East, which is it's sort of an endemic virus also. So it's hard to know. When you look at many of the models about COVID-19 that exist, and there are hundreds of them, many people see COVID-19 as being a recurrent infection that assumes a seasonality, but is just more lethal, if you will, than the regular seasonal coronaviruses.
Right.
My guess would be is that it's you know, a lot is going to happen in terms of selection pressure when we immunize people, right? That the coronavirus will come up against a significant proportion of people who have protective immunity. And the question will be is, the coronavirus will do everything it can to make sure that it can still infect people. And so there will be a tremendous selection pressure for the coronavirus to mutate. And maybe those mutations will be beneficial for us and that it'll disappear. But maybe those mutations will not be very beneficial to us, and it'll transform into something much more lethal or something that can evade our protective immune responses, requiring us to then develop yet another vaccine against a newly emergent strain of coronavirus. So it's a great question, but I mean, nobody could, I can speculate as anybody would be speculating about what might happen.
Okay, thank you.
Actually,
I
I have a
guess.
it's Sue Jones. I have a follow-up question to that. Do you foresee a vaccine like this becoming more of a standard like the pneumovax for people over the age of 60, say?
Like a coronavirus vaccine? Like just in general, or do you mean like the mRNA vaccine?
Like in general, if this
General.
continues and the disease does not just disappear,
Yeah.
could you see this being kind of one of the vaccines that people get over the age of 60?
Yeah, well, there's already talk in the literature about wouldn't it be great if we could develop a combination influenza coronavirus vaccine? So people are already discussing that and looking into the future and saying that this will probably be the reality.
Okay, good. Thanks.
Unmuted.
No, you're unmuted.
Giulio, it's Bob Lewis Watts here. Thank you. That was a really excellent talk. I had a question. There are, in the past anyways, that have been flu vaccines that have been given intranasally. Is anyone working on that for SARS-CoV-2?
No, I don't. I shouldn't say that. Let me go back to my WHO slide. I'm not sure what proportion of the vaccines in clinical production are live attenuated. So 1. So of the 61 in clinical development, only 1 is a live attenuated virus. So obviously not very many.
Okay, thank you.
Kelly, if you're talking, I see your lips moving, but I don't hear you.
There, can you hear me now?
Yeah.
So what's going on then in the UK? What is this news that there's this new strain that's 70% more spreadable or contagious? 70%.
Yeah, it's not a new strain. Can you see the slide, the map?
Yeah.
Yeah. So if you look at, this is the ancestral virus, and here's the mutation. Okay. You can see here, there's a little yellow, tiny little yellow circle in there. So it's not that it's necessarily new. I think it existed already. It's just sort of spread more globally, where now it's the predominant strain where people are looking for it, right? Where people aren't looking for it, it's probably there anyway. They just haven't detected it. I mean, it's, as I said, it appears to confer greater infectivity, which probably explains why it's globally disseminated. But the mutation, as I talked about, is not in the receptor binding domain of the spike glycoprotein. So it's highly unlikely it's going to have an impact on vaccine efficacy. And certainly in vitro, we've already shown that the convalescent sera of infected patients with native virus is still able to block the entry of the mutant virus into human cells. So it appears as though it can't evade our immune system. So in theory, then it should not result in more severe disease. And there's no reports that people who are infected with this D614G strain are in fact much more likely to get sick or have more severe disease or die.
No. Okay. Thank you. All right. Any other questions? We can let Giulio go back to work.
Thanks very much, everyone.
Okay, thank you very much, Giulio. This is very informative.
Cheers.
Thank you. Stay well.
Bye.